Hello, my name's Samantha Self, and today I'll be reviewing the topic of memory with you. Essentially, we will be reviewing our current understanding of memory, how it works, and what about when it goes wrong. So, we'll review learning and memory, memory storage and amnesia. I'll discuss amnesia in animal studies and discuss a bit about it in humans too. There's so much to learn about memory. There's so much I learned about memory. I mean, who would we even be without our memories? So let's dive in. The first article I think we should go over is called Activity in Both Hippocampus and Parahynal Cortex Predicts the Memory Strength of Subsequently Remembered Information, authored by these lovely people in 2008. Um, So basically, this article reviewed the parts of our brain that are active when we're learning something and how that correlates to memory strength and our recollection. So they used studies looking at cortical regions of brains that are lit up during learning or even during mind wandering times and in recall. So they used fMRIs to look at the brain in their studies. They'd give people a list of items to study while being scanned and then gave them a recognition memory test outside of the scanner. It's kind of like seeing what their brain encodes and then retrieves. They compared what parts of the brain were active during studying in correlation to what was remembered. So they looked at the hippocampus because that's where they've known recognition based on recollection to stem from, which involves remembering specific contextual details about a previous learning episode, which is what they were doing before when they were trying to give it items of things to remember. They were in their learning episode. While parietal cortex has been associated with being important for recollection based on familiarity, which, as the article says, involves knowing that an item was presented without having available any additional information about the learning episode. Also, during their test of recognition, the people taking the test gave confidence ratings to old and new words on a 6-point scale. They didn't make any other judgments whether their decision was based on recollection or familiarity, and you can see that here. So some of the words that were in code, some of the ones that were remembered and retrieved, confidence ratings given for words. Um, so interestingly, when some parts of the brain were more active, parts which may be referred to as the default network, people seem to appear to forget more items they studied. Some of these regions are known to be activated when the mind is wandering and when there are lapses in attention. So I thought that that was interesting. I would think our brain just wouldn't be active if our mind's wandering. Well, I guess not completely active, but I mean, not active, but anyways, there's areas specifically that are lit up when our mind's kind of wandering and we're having lapses in attention. So when these were more active in the scanner, such as the bilateral inferior parietal cortex and the posterior midline, the participants actually remembered less of the items they were supposed to. So this mind-wandering activity could lead to issues with memory encoding. However, other parts that were active, such as the left inferior frontal gyrus, predicted memory success, and people seemed to remember more when those parts of the brain were active, as well as the temporopolar cortex, the parahynal cortex, and the hippocampus activity. They all correlated with a stronger ability to remember studied items. So that's kind of some of the areas in here. This right here is where like kind of lighting up the hippocampus. Um, just in case you didn't know that. So they noted how the hippocampus and the parietal activity correlated with a stronger memory for remembered items. So I thought that was pretty interesting. So it looks like a lot of our memory is held in that area, at least for these memories. So the next article review is called The Biology of Memory, a 40-year perspective, kind of like a history of a lot of the things we've learned about types of memories, authored by Eric Kandel in 2009. So the go withdrawal reflex, which I was like, what? I had to research that. The go withdrawal reflex of aplasia is a study of sea hares or sea slugs or and their defensive reflex. So we can study, they have a really well-defined neural network, especially in relation to how they learn, which we can study, which is reflected in their memory and in relation how we learn in our memory, and we can study their habituation, their sensitization, and more. The article also reviewed what people have learned from rats and from flies. It was basically a lot of stuff about animal studies and what we've learned from animal studies. So basically, the article discusses the biology of memory, both declarative, like episodic memory, and procedural. They go over what we've learned. So to get some review on this, the medial temporal lobe, which contains hippocampus, encodes episodic memory, which is memory for events, episodes. It's autobiographical. It's a type of declarative memory, so we can talk about things like experiences we've had. It's tied into time. It allows us to relive past experiences, and all our sensory cortical areas have projections to the hippocampus. When you see something, the neurons that encode and produce your ability to see what you're looking at are sending action potentials through their connections to produce activity in the hippocampus. The hippocampus produces activity, which then sends projections back into the cortex, which can cause activity in the differing sensory cortical areas and emotional areas. This is all important for the processes of encoding and retrieving through the hippocampus in regards to episodic memories, which is like declarative memories. And that replay mostly tends to happen while we're sleeping, so get good sleep. And then the long-term potentiation will strengthen the synapses of neurons that fire together in time, because neurons that fire together wire together. And long-term depression will weaken the connections, which will weaken your memory. The article also discusses procedural memory, which is also known as motor memory. It's implicit, it's non-declarative, it's different. It's like a how-to memory. It's like how you remember to ride a bike, you know. Even being depressed can be a procedural memory. It's like something that your brain just remembers how to do. It's stored in the putamen, the cerebellum, and the caudate nucleus. So if you get any damage there, kind of damages your procedural memory. You might struggle to walk or to play an instrument. The article also reviews synaptic plasticity. So that's kind of the process of how the strength of connections between neurons change. It's a big part of our learning and memory. The article actually reviews a lot. I don't think I'll have time to get into it all today, so I chose what I found most interesting, and I think would be most helpful to learn. So, anyways, this article found, as I quote, studies of the go withdrawal reflex also reveal that even elementary forms of learning have distinct short-term and long-term stages of memory storage. Whereas one training trial gives rise to a short-term memory lasts minutes, repeated spaced training gives rises to long-term memory lasting days to weeks. So they were able to study this by poking and prodding with the animal, which is actually kind of sad, but they could see how it learned to react to the stimulus. Anyways, and this has been proven true with us. Spaced repetition has proven to be more effective in our learning. They also found that enhanced synaptic plasticity and learning in the hippocampus shows that synaptic strength is important in memory formation. Oh, and looking at the EEGs on mice, they found that recently stored spatial memory is often conveyed to the neocortex during sleep, kind of like I said before. So, and today we've learned that this is true in humans, and disruptions of sharp wave ripples we experience in REM sleep causes worse memory consolidation. So, excuse me, better sleep equals better memories. Anyways, I could be here forever if we talked about this article. It's pretty interesting. But I really started getting interested in the anterograde and retrograde amnesia that we were learning about, and kind of like just the psychology and biology behind that. So let's look at it in rats. Anterograde and retrograde amnesia after lesions to the frontal cortex in rats, authored by these lovely people. It's a pretty interesting topic. So you might be asking, what is retrograde and anterograde amnesia? Well, anterograde amnesia typically it results from an injury to our brain, and it's when an individual doesn't have the ability or has an impaired ability to form new memories after the injury. They do have a good memory of everything that happened prior to the injury, though. And in retrograde amnesia, after an injury, the person has no or impaired events prior to the injury. So, however, there's no issues in making new memories for them. Both crazy, both sad. Not crazy, but an interesting thing that happens, and sad if it were to happen to anybody. Um, Although pure retrograde amnesia is pretty rare, it usually does kind of co-occur with anterograde amnesia, which is also actually something we're going to be talking about. So we'll look at this in rats using food preference studies, which I'll get into a little bit. So basically, rats were presented with a few options of what to eat, and they developed a food preference before having surgery to cause hippocampal lesions, causing retrograde amnesia. Once the surgery happened and they were presented with the food again, they immediately showed no preference for the previous food. This is kind of showing where they had the lesions in their brain. Anyways, it seems that they didn't remember that it was the food they preferred. So, yeah, and then also when the rats were given frontal cortex lesions to incite anterograde amnesia, they at first had the same food preference. However, with more time, they seemed to not anymore. So that was kind of interesting. The researchers also ended up killing many of the rats and examining their brains. So, hi, ethical dilemmas. I don't think that sounds great, you know? But as said in the article, an important finding of the present research was that anterograde amnesia at long retention intervals and retrograde were correlated in rats with frontal cortex damage. So just the frontal cortex damage to both really showed that there was a lot of intertwining. So this showed the researchers that at long delays in rats, amnesia was very intertwined with retrograde amnesia. But all of this doesn't just apply to the study of rats. So the article covered how it and others have confirmed that specific types of brain damage can cause cognitive deficits that affect both anterograde and retrograde memory functions. So hippocampal lesions that make consolidation processes of memories difficult do cause significant anterograde amnesia and often retrograde amnesia in rats and us. So we should be trying to keep our hippocampuses lesion free, or just not really get any injuries there, so that we don't start forgetting everything and our synapses stay strong and connected, and yeah, we don't really lose our memories. So I was just fascinated by everything on the topic of memory. I was interested in the encoding and retrieval of it. I think there's a lot of ethical dilemmas in how much research is being done on the animals that are being tested. Also, there's some worries there because animals aren't humans, and I think there's a lot of difficulties in this topic of research, considering the fact that we're not going to like poke and prod our fellow humans and then like dissect a human brain during the study, to look at it. So there's a lot of ethical dilemmas in this. I do think a lot more research should be done so we can know more about amnesia too, the retrograde and anterograde. There are some other interesting articles about it, and some good things we learned in class about it. I think if there's something that we should study further, it's how memory should be researched more in consideration to eyewitness testimony. I mean, I just want to know like the biology behind our memory even more, and how that can like maybe how our emotions and our biases can shape our memory, so that maybe if we are going into like an eyewitness situation, we have to understand that it might not always be accurate. I don't know. That was just a an interesting thought I had. But I think there's so many things to continue to learn about in regards to memory. Um, our brains are wild, and we wouldn't be who we are without our memories. So these are my references, and thanks so much for tuning in to my overview of a couple research articles and some hopes for continued studies on the topic of memory. Thanks.